revision. And the topic itself is the cell biology topic. Now these are then the instructions, right? So firstly you need to watch this video, which is a sessions instructions video, right? At the end of the video there's a number of questions, right? And what you need to do is you need to write down the answers to the questions right from this video into your revision book. You write out the question and then you write out the key or the sentences afterwards. In the answers you must use the keywords and it must be in a logical sequence. Now the best way of actually finding the answers themselves is there's another video that's called the walkthrough or reverse the classroom video for cell biology. And just for any additional work, if you then want to even improve even better, right, watch the knowledge test video, which just has questions on it. And what you do from that is then you write down your answers onto a piece of paper. And then the final one is you can then watch the same video but with all the answers in it to make sure you understand exactly where you got wrong.
So the first session, you've got 5 activities to carry out. The first session is to compare a prokaryotic cell with a eukaryotic cell. Now you also need to include a diagram of an animal, plant, bacterial cell, and you need to label them fully. Now you've got your keywords, which are nucleus, cytoplasm, cell membrane, cell wall, ribosomes, mitochondria, chloroplast, vacuole, flagellum, slime capsule, and plasmid. Now you'll find that you'll not need to use all those labels with all 3 types of cell, but you need to label each one fully and explain what each part does. Now we don't want you to spend too much time on drawing the actual cells. So for instance, it doesn't take very long to draw an animal cell. It's taken me about 10 seconds to do it. We've got 5 labels on it, just to give you a reminder, and that is the kind of diagram we're expecting to have in your exercise books. You're going to do that for the other 2 types of cells as well. The second activity is for you to describe the method for calculating magnification, and you also need to remember to explain how to convert the units. Third activity is to describe the cell cycle, and the instruction is to include the pie chart. Now the pie chart, again, shouldn't take you long to draw a circle, and there's 3 parts to the cell cycle. That's what we're looking for, but you need to label it and explain what is happening at each of the 3 stages. So the keywords you need to include are organelles, mitosis, cytokinesis, chromosome, and DNA. The fourth activity is to describe the 3 methods of transport in cells, and they are diffusion, osmosis, and active transport. It could be a good way to compare them by drawing a table. It's up to you the format that you take, but you need to include in your answer about particles, concentration, water, partially permeable membrane, passive, energy, and concentration gradient. And then the final activity for this session is for you to compare how embryonic stem cells and adult stem cells can be used to treat disease. So your keywords are differentiate and therapeutic cloning. You need to complete this in your green exercise books, and you need to bring this to your next science lesson where your science teacher will be checking your work.